We ready? Well, it is Friday. So we need to think about part 2 of the material. So the outline, excuse me, the Word document that's posted in EduCat now is the complete outline. I put everything on there now. So if you download that, you'll see that part 2 is on there. And so the stuff we haven't covered yet starts with this material, looking at the carbon fixation ideas. So, of course, the C3 cycle, pretty important. I think one of the key slides to look at is the pie chart. I think that's a good one to study off of, because that gives you a lot of the big picture ideas in one fell swoop. So you have the different stages: carboxylation, reduction, regeneration, and then the key, like what starts it off. And the only thing they really don't put on here is that's of course very important, is the Rubisco enzyme. So make sure you are familiar with the first step, including the enzyme. And then, like, what do you end up with at the end of each phase here? So at the end of the first step, you have PGA. At the end of reduction, you have G3P. And then, of course, at the end of regeneration, you're back to RUBP. So big picture ideas, but thinking about Rubisco as well. And then the other thing too they show on here is looking at where are the NADPH and ATP products being used. So of course the light reactions tie into what's happening with the Calvin cycle, because the products of the light reactions, the ATP and NADPH, feed into the Calvin cycle that's cooking along in the stroma of the chloroplast. Any questions on C3? And then we got into the PCO, that's kind of a mess. Again, big picture ideas for the final, thinking about the very first step is really critical, so that you should know. So looking at, let me put not sure which one you studied off of, but let's put this one up. And so here with the RUBP, reacting with oxygen instead of with carbon dioxide. And so Rubisco is to blame for that one. That enzyme is a little bit sloppy. And so that kicks off the PCO. So thinking about the very first reaction, you're looking on the left figure, it's on the top left again as well, RUBP and oxygen. And you get phosphoglycolate, which has to go through all of the PCO twice, essentially. The other product, PGA, can immediately feed into the Calvin cycle. So knowing the first step, what's going on to kick that off, and then the organelles involved, and the fact that you have extra energy, it's a cost, not for free, but going into the details of, I won't be doing that. Okay. So essentially what starts it off, what do you end up with, what's the whole payoff, but not all the intermediate steps. Okay, and then we looked at C4 and CAM and how they're able to create a situation where they don't have this happening. Or if it does, it happens very infrequently and very modest levels. And so the C4 and CAM are designed to really flood Rubisco with CO2, basically. And so we looked at C4, And again, you know, the first step is something you should know, what kicks it off, what enzymes are involved. And then like the different stages. So we talk about carboxylation is the first step. And then the decarboxylation is actually the second step when you have the malate coming in and moving the bundle sheet. That's your decarboxylation. That just means you're decarboxylating malate is what they're referring to there. So the decarboxylation, malate shifts over, bundle sheet, gets cleaned off of it. And then the regeneration step is to get that pyruvate back so you can make more PEP. So again, the point here is when you look at the bundle sheet, that CO2 is saying that feeds directly into the Calvin cycle. So you have to imagine Rubisco sitting there. And Rubisco is just getting all that lovely CO2 that's coming off of malate. So the CAM and C4, you know, a lot of the same players in the pathways, but you know, this is a spatial situation. They spatially segregate where certain things are happening. And then the CAM is a temporal day-night rhythm for when things are happening. So CAM compared to C4. Here we have the CAM idea. It's all happening in the same cell. You don't have that physical separation, but you certainly do have what's going on at night versus what's going on during the day. Any questions on the C4 and CAM? All right. Well, then we got into the next slide set. We were looking at sunlight and how plants respond to sunlight. So for that one, I want you to think about photoreceptors again. So the definition of photoreceptors. And then we really only talked about a few here. So the cryptochrome and phototropin for your blue light. I gave those examples, and then the UVB receptors, the photoreceptors, the UVR8 for that, and then phytochrome. So of all of those, I'll certainly ask more about phytochrome than any of the others. So do we have questions on phytochrome? So there was a question on the previous exam that we just took where it was asking about if a plant senses it has a lot of PSAR, then it knows it's in sunlight, true or false? Or is it the other one? PFR? Right. So if a plant senses it has a lot of PSAR, what is creating a lot of PSAR to build up in its body? Fire. So I know it's confusing. I get it. I had to grapple with this a lot when I was an undergrad, and so it's a strange concept. And I wish they would have named it something. I don't know. Maybe if they had named it, but they didn't. So when a plant has a lot of PSAR, that's saying it's getting hit with a lot of far red light. It's getting hit with a lot of far red light as opposed to red light. That means it's probably in a dim, shady condition. Because if a plant had its choice, its top pick, it would be, I want red light. And red light comes from sun. And if you are in the upper canopy, in a forest system, that's what you'd do. You'd hog all the red light because that's going to be your big payoff for photosynthesis, all types of growth, flowering, and all of that. So if a plant has a lot of P(R), that means they're getting far red light exposure over red light. So that would have been a false statement. They don't know they're in light. It's just the opposite. They're like, oh, I'm being shaded if they have a lot of P(R). So it's this weird, you have to really think through what's allowing for P(R) to be high levels of that versus P(F). Yes. Not to the point of like boosting off an electron or anything like that. Yeah, I think I know which one you're referring to. Yeah, so and that's going back to the definition of a photoreceptor. When you think about a photoreceptor, they don't get excited and boost off electrons. They just absorb whatever wavelength of light they're adapted to, and then once they do that, they shift their physical three-dimensional shape. So that's kind of the core thing about these photoreceptors. And so nowhere on these diagrams do they show an electron getting shot out with some kind of acceptor molecule. You don't have that happening. But what does happen is that in this case, you're seeing the cytochrome absorbing red light, shifts into the so-called active form. And what they're trying to say here is that it's that form that will actually interact with other proteins that then govern gene expression, that will control gene expression in some manner. Maybe it's going to tell the plant, go ahead and start flowering, perhaps. So it's kind of funky that it's just a shape change that's allowing for that version of cytochrome to then send the signal, interact with these other proteins, and then get stuff done. It makes things happen just by changing shape. It's kind of a really simple idea, but it works. Other questions on this slide set? Okay, well, the next one is looking at signal transduction and hormones. Yes. So that's looking at this photoreceptor. So the UVR8 is a photoreceptor, and it is the one that they've discovered that will absorb ultraviolet UVB light. So if, part of the sunlight coming down to Earth is, of course, ultraviolet and UVA, UVB. So they said UVB portion of ultraviolet light that will cause this photoreceptor to actually they break apart into monomers. So that's a little bit more drastic than just changing shape. It's actually break apart. So that was kind of interesting. But anyways, it's just this UVR8 photoreceptor absorbs UV light. That breaking into monomers will then allow, that's like the physical way that then that UVR8 will interact with proteins and shift gene expression. And so it's just a way for a plant to assess, like, oh, I'm getting hit with a fair bit of UV light. And then they might respond by maybe shifting their leaf orientation so that it's, the sunlight starts to spill off a little bit more. Yeah, we'll go back into a dimer. Yep. Kind of funky. Okay, signals and signal transduction. So there, signals and signal transduction. So figure 12.2 is this one. So this is the big picture idea of what happens in the signal transduction process. So the information that you see in the little short boxes here. So you get some kind of signal that has to dock into a receptor, and then you'll get probably some level of amplification of that signal in some manner, like second messengers. And then you have to get that signal transmitted, usually all the way to the DNA to shift gene expression in some way. So the basic idea. And then 12.3 is the next figure. This is getting across the idea that there's, dedicated receptors for different hormones in this case. And they're found throughout the cell. They're not all embedded in the cell membrane. You can find them in different membranes inside as well. But certainly each one has their own dedicated receptors throughout the cell. And then second messengers. Here, this is the next slide that shows the list. So those are the ones that I want you to think about. There's more than that, but this is what we'll focus on. So what are second messengers and who are they? So that's the list. And then we talked about a fair bit of different hormones. I'm going to just zoom in on the classic 5 for this slide set, where we're looking at the auxins, cytokinins, gibberellins, abscisic acid, and ethylene. So I say key characteristics and uses. So for example, like with auxins, I won't be asking any chemical structures, but some of like the big ideas, looking at, this is a key finding that it can slow down the stem of a tissue, and or the tissue of a stem, and if you shine unidirectional light from the side, it'll shift over to the dark side. That was one of the earliest findings, actually, that Darwin was able to demonstrate. And so again, with this, if I ask about the hormones, I will pose a question that has several traits listed. This hormone does this, 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 and this. What is it? And then have options. I might end up organizing that as a matching or drag and drop. Matching, maybe? I haven't finished taking the exam yet. But I will, with the classic 5, I will be asking like key things that they can do for the plant. So hopefully between what you've been learning in lecture, the labs you did on hormones, some of that will sound like repeats. It'll sound familiar. Let's see. So this slide set had, come on. So this slide set had the classic 5, and then, like, knowing that ethylene is the gaseous one of the classic 5. And then we did get into some of these others too. I'm not going to really talk about them in any detail, the steroids or spironolactones. And then if I talk about salicylic or jasmonic acid, it would be more dealing with defense, how they defend themselves. So also thinking about, where is it? Well, I guess that's more in mechanisms. Questions on anything in this slide set? The next slide set does have, oh, where'd it go? Applied uses of hormones is part of, no, I didn't put that specifically on there. I'm gonna have to amend the outline because the applied uses of the hormones does have complementary information on the hormones, of course. So when I'm asking questions about the classic 5, some of it will come from signal transduction slide set, and some of it will come from the applied uses. So I need to amend the Word document. Well, I guess it is there. So number 11 on the Word document outline, it does say both of them, signals and signal transduction applied use of the hormones. So if I ask, like, this hormone does this, this, and this, it would come from information off of this slide set and then the signal transduction slide set. Anything on here you need clarification on? It'll be similar to that. Yeah, that part will be similar. Kind of big picture ideas on each of the classic 5. And I'll try to give enough info in the question that at least hopefully a couple of the ideas you'll get a feel for what I'm talking about. Anything on here? I heard the plant tissue culture results went pretty well. That's sweet. I'm really happy. We've had issues in the past with contamination that's not always due to the students. It's something like with endophytes, you can't really, there's not anything you can do about that. So you guys did a good job. Tissue culture. That was nice to hear that there were so many successes. Well, the next one, slide set, is mechanisms of, or secondary metabolites, sorry. Secondary metabolites, now we're thinking about the big 3 categories: terpenes, phenolics, and nitrogen containing. And so, at least so far, the terpenes are the biggest category. And of the 3, I think I have it on here, of the 3, I'm going to ask more about the nitrogen containing metabolites than the other 2. So that's toward the end of this slide set, where we get into the nitrogen containing compounds, slide 21, where we're thinking about, the reason I'm kind of focusing on them is they're the ones that greatly impact mammals and have been adapted to interact with mammals. So. And then endophytes, again, very broad, basic idea on endophytes. So not a whole lot coming from that slide set, really. And then the mechanisms of defense, I list out some figures that would be really good to study. So figure 18.14, this kind of has a lot of the big picture idea all in one fell swoop, where you're looking at the caterpillar chewing on this lower leaf, and they're trying to say how that kicks in this whole cascade of the plant picks up on something in the saliva of the caterpillar, and that's going to cause the cysteamine polypeptide hormone to dock into its receptor, which kicks off the signal cascade that allows for jasmonic acid, that hormone, to get made. And then the beauty of that hormone is that it can flow to distant leaves and tell those leaves to ramp up defenses. And so it's a very nice ability for the plant to systemically warn itself. So we talked about that it's not just hormones that can do that. We have electrical signals, vibrational signals, volatile signals. It's not just always through the phloem, or for example. Figure 18.15. It's another one on the word outline. So again, just thinking about all the different ways that they can send the signal systemically. And then figure 18.16. No, is that the one? 18 point, where's 18.16? Oh, that one. There's 18.16. Again, the volatile idea that you have the capability of warning nearby plants, or even recruiting some of the predators to the pest that's attacking you, which is pretty slick strategy. So that's a nice kind of big picture idea slide. And then this one I also put on the list. 1819. So this is comparing MAMP versus effector triggered immunity. And so it's a nice visual showing some differences here of how it's working, where you have these receptors, the PRR receptors, pattern recognition receptors in the membrane that will detect usually any damage that's being done or bits and pieces of the pathogen. And then that triggers the MAMP triggered immunity. With the effector, that means that there's something coming into the cell. Toxin, perhaps. It's often a toxin that they inject into the cell. And then you have these R proteins that are swarming around in the cytoplasm that are trying to defend against that. Because as soon as they dock with the effector, whatever that's coming from the pathogen, that's going to trigger their effector triggered immunity. And so it's kind of a 1-2 punch. It's the plant's ability to have different mechanisms in play, so that if they call this the first line of defense with the MAMP that failed, hopefully the effector triggered can nip it in the bud still and not have a full-blown infection or attack. So that's a good visual. This one, figure 1820. Again, this is a very big picture overview of different strategies that the plant can do. And so the plant will actually upregulate its reactive oxygen species, which is actually a good thing. That's capable of allowing the plant to defend itself, fight back. It's kicking in signaling pathways to allow for some of these other things to happen that you see in the white box. And so big picture idea of, what's the hypersensitive response versus cytoalexins. So knowing the big picture ideas of these defense responses. And then systemic acquired, again, is different from induced systemic. So keeping those two clear. So it's, knowing, like hypersensitive, that's the one where you get a lot of local cell death, right where the pathogen is. So the plant will sacrifice, essentially tell those cells to commit suicide right where the pathogen is to try to limit spread. It tells those cells to commit suicide, and then it sucks away all the nutrients away from the pathogen. So it's just trying to kind of create a void so that the pathogen is kind of starved of nutrients. If you want to think of it that way. And then often what they'll do after they do that is some of these cells will ramp up in their cell wall structure to try to kind of create a wall, so to speak, to limit spread. And then when you think about phytoalexins, those are just secondary products that are really very strong and very capable of having very potent killing ability against a lot of different pathogens, fungi and bacteria. And then lignin is just, you know, trying to strengthen its walls, so to speak, trying to just ramp up its physical integrity to keep the pathogen from spreading. And then the hydrolytic enzyme, that's just saying, you know, the plant's going, oh, I'm getting attacked by a fungus. I'm going to start ramping up a bunch of chitinase enzymes so I can attack the fungal tissue, you know, because fungi have a lot of chitin, just like insects have a lot of chitin. And so the biosynthesis of enzymes, that's the plant starting to make key enzymes that will attack that particular pathogen and try to actually break down the pathogen, physically break it down. Any questions on what's going on in this slide? What about ISR and SAR differences? Which one is due to the plant kicking off a defense response, a systemic defense response? Which one is directly through the plant's action? Yeah, SAR. So the induced is where you have an outside entity that's triggering the plant. It's inducing the plant. Hey, you know, heads up. You have a pathogen lurking. And so most of the research has been done with bacteria, but they now know that fungi can do this too. So it's a coexisting beneficial microbe hanging out with the plant, and they can sense that something's going on, and they will send out a signal to the plant that gets the plant to ramp up against that particular pathogen. So try to keep those two, the big picture idea, separate. ISR is induced from an outside entity, some bacteria or fungus hanging out with the plant. Whereas systemic acquired, it's the plant going, oh, I'm getting chewed on, or a fungus is attacking me. I know what to do. And so it's the plant directly assessing. And then really the abiotic stresses, there's not a whole lot of content there. It'll probably look similar to what we already asked, because I didn't really get into too much detail on these different slide sets. We're just looking at chilling, drought, and flooding. So there's some, you know, what can the plant do to try to deal with it, try to stop it from impacting them too much. So thinking about what hormones are involved to help them. Drought makes sense to try to devote growth into roots for drought so they can search and forage for more water. And then close your stomata. That's obviously a good idea too if you're in a drought phase. So again, just thinking about like what are some key ideas on how would the plant deal with drought conditions to try to survive it. Shifting their leaf morphology is pretty common. Even their orientation to the sun to try to spill off the sunlight so they're not getting too much sun. So they're capable of, on the fly, they can start shifting their morphology. They don't have to wait, you know, it's the same plant living. They don't have to like pass it on to the seed and into the next generation. So if the plant is starting to experience that drought, the new leaves that come out, they might be smaller, they might have more trichomes, and it's amazing. They can adapt on the fly like that. And then with the flooding, we have a little bit, we have what's happening with the plant in the sense that the plant is now going to be limited in oxygen because anytime you have the soil saturated with water, oxygen just doesn't like to diffuse very quickly through water. And so the plants, the roots start to get starved of oxygen. So you have that whole issue. Obviously that's a really bad thing. And then you also have microbes in the soil. Some of the microbes in the soil that are anaerobic, they start to really build up in population, and they start doing all kinds of stuff that's not so good for the plant. So there's a little bit more going on with the flooding idea in terms of the associated microbes. And then when you're flood, this is the opposite rather than with a drought where the roots will focus on growth. With a flood, it's just the opposite. Now the stem will focus on growth, try to keep their head above water, so to speak. And that's, again, they can do that on the fly. They'll start to elongate their stem. Okay, so what else? Any questions from any of the slide sets? Yeah, again, they have to name it similar, right? So the jargon, I tell you, scientists love jargon. Okay, so with this idea, with the C4 and the CAM cycle, so this is the C4. So what kicks it off to start with is the CO2 comes in and it's going to react with phosphoenolpyruvate. So that's PEP. So that is the 3-carbon molecule that's going to get the carbon dioxide attached to it. PEPcase, on the other hand, stands for PEP carboxylase. So if you can remember, I would probably put it on the exam if I probably put it as PEPcase. That's probably how I'll have it if you visually. So PEPcase, but again, the ACE on the end of that case, ACE. Anytime you see that in science, that means it's an enzyme. So if you can remember that, and so what's happening is that PEP will come into the active site of PEPcase, CO2 comes into the active site, and then that allows the PEP and the CO2 to react. And so PEPcase is great because it doesn't care one bit about oxygen. Oxygen isn't going to be able to get in there and react with the CO2. So you greatly limit any issues with that. And then that combined with the idea that rubisco spinning the bundle sheath cell, it's kind of physically separated from a lot of oxygen exposure. Now it's getting bathed as well. So this spatial segregation and the use of PEPcase really does a good job of making sure rubisco is not seeing oxygen. It's just seeing all that lovely carbon dioxide. So that's happening in both the C4 and CAM. It's just here they're showing the same thing. where they're showing phosphorylpyruvate and carbon dioxide reacting in the active site of the peptase enzyme. I haven't finished making the exam. I still think it's going to be somewhere between 80 and 100 points. If I had to guess, I'd say closer to 100 than 80, but I can send out an announcement when I finally have it final draft. I know some people were asking about that because they're trying to play the math game of what grade, like they're trying to figure out what their final grade's going to be, and knowing how many points the final is important. So I get that. I'm trying to finish it off, and then I'll get a message out to you. And it's just, all multiple choice and matching maybe, and no essay essentially. It's just going to be one exam that you'll click on. And it's same idea, ask questions. If I can get you thinking, get the right nerve cells firing, let me help you. So the final exam is what, Wednesday, right? Wednesday at noon in this class. And so between now and then, including the weekend, this weekend, if you have questions, send me an email. If you want to meet with me next week in person, great, we can do that. Especially because next, week there's no classes, so my schedule is much more flexible. So if you want to meet in person too, yeah, absolutely. We have till Wednesday till noon. So I'll be checking email regularly through the weekend. Other questions on any of the slide sets? Or anything else? For the final? Yeah. No, I could do that and see if there's a clear ranking. Yeah. Yeah, I knew there was going to be a clear ranking on this previous exam, because I knew that like the flooding stuff and drought and all that wouldn't be very much. And I knew like the mechanisms of defense, so I wanted to strategize that for you. So I can do that again. Yeah. Okay. Yeah. Yeah, especially for a final. And I think that would be helpful. So um a cumulative final, that would make sense. So once I get that all finalized, I'll be sending out an announcement. I don't know if I'll get it done before Monday, but I'm gonna try. I'll make a note so I don't forget. Any other questions? The other thing, if you could please remember to do the student evaluations. You could even do them right now. If you want, I can leave. I'll be leaving here shortly. I'm not supposed to be in the room when you do them. But if you could just spend a couple minutes to do those, that'd be great. So it's on the EduCap page. Student ratings, I think, is how they phrase it at the top of the page there, EduCap page. Okay. Hang in there. We're close. Make sure to ask, send me emails or set up an appointment. And then I'll be feeding you out information as I get things finalized.